Characterization of bacterial culture in agar plates. We study characteristics and morphology of bacterial culture to describe bacterial structure, to determine bacterial species, to distinguish how various growth media will affect colony growth, and to differentiate between gram-positive and gram-negative bacteria. What is microbial colony? Microbial colony is a visible cluster or mass of microorganisms growing on the surface of or within a solid medium, originating from a single cell. For the colony morphology, it is a method that scientists use to describe the characteristics of an individual colony of bacteria growing on agar in a petri dish. Even though colonies belong to one species, they may differ in their shape, size, color, and texture from others. The following are the main characteristics of the morphology of a bacterial colony. First, we have color, surface, shape, size, margin, and elevation. These are the different descriptions of each characteristics of bacterial colony. Pigmentation of the colony. Some bacteria produce pigments when they grow in the medium, like the green pigment produced by Pseudomonas aeruginosa, the buff-colored colonies of Mycobacterium tuberculosis, and red-colored colonies of Serratia marcescens. This is what Pseudomonas aeruginosa looks like in an agar plate. Pseudomonas aeruginosa is the most often causes infections in humans, in which it causes infections in the blood, in the lungs, or other parts of the body after surgery. Here are some examples of colonies pigmentation. Surface characteristic. There are different surface characteristics of a colony. First, we have smooth, we have glistening, we have rough, wrinkled, dry, powdery, radiate, concentric, contoured, and many more. Here are some surface characteristics of a colony in an agar plate. First, we have glossy, we have slimy, we have dry, and we have smooth. This is an example of Nocardia africana, in which it has a dry with powdery surface. Shape and form. There are so many shape and form of a bacterial colony in an agar plate. I'll give some example on what they are look like. First, we have circular, we have irregular, and we have rhizoid. Size of colonies. The size of the colony is determined by its diameter and measured in millimeter. Knowing the size of the colony can be useful characteristics for identification. The terms to describe the size of the colony are pinpoint, small, medium, and large. Here are some examples of colonies that can be found in the blood agar plate, in which the first colony that we can identify has 2 millimeter in diameter. The second colony, as you can see, it ranges around 3 to 6 millimeter in diameter.
The fifth characterization of bacterial colonies is margins. The first one there is the entire margin, which has clear and notchless areas. Number 2 is the undulate with wavy and shallow indentations. Next is the lobate with wavy and deep indentations. Finally, there is the curled with parallel chains in wavy strands. I have also included a number of other margins of bacterial colonies below. The sixth characterization of bacterial colonies is called elevation. In the first picture, the area labeled with the number 1 is called a raised elevation, and it has thick and abrupt edges. The one labeled with number 2 has no elevation whatsoever is called a flat. Up next is the convex elevation, which has a segment of a circle but flattened. Essentially, it is shaped like a semicircle. Then the last elevation is the umbonate, which has raised but with thicker center than margins. I have also included a number of other elevations not shown in pictures. The seventh characterization of bacterial colonies is odor or scent. The first one is the Pseudomonas aeruginosa, which produces a fruity, grape-like scent. Number 2, the Streptococcus anginosus produces a caramelly, pastry-like butterscotch odor. The third one is the Moxella catarrhalis, which produces a peculiar semen-like odor. The fourth one is the Echinella corrodens, which produces a familiar scent for cleaning, which gives off a bleach-like smell. And then the last one is the Proteus mirabilis, which produces a rancid rotten chicken soup or broth scent. The last characterization for bacterial colonies is called the pigmentation. Essentially, it is how the species spreads pigment to the medium. And the first one is the Pseudomonas aeruginosa, which has green pigments. The next one is the Mycobacterium tuberculosis, which produces a buff-colored colony. And then the last one, the third one, is the Serratia marcensis, which produces a red-colored colony.
Characterization of bacterial culture nutrient broth. Nutrient broth is a liquid medium used for the cultivation of a wide variety of organisms from clinical specimens and other materials. This medium can be enriched with other ingredients such as blood. Surface growth. Ring at upper margin adherent to glass. Pellicle growth in the form of a continuous or uninterrupted sheet. Some bacteria form a layer of growth across the top of the broth in a tube. This layer of growth is called a pellicle and correlates with strict aerobes since the greatest amount of oxygen is found. Flocculent, small masses of growth of various shapes floating in the culture fluid. Some bacteria form clusters of growth that are either floating in the broth or stuck to sides of the tube at every level. From top to bottom, these bacteria correlate with facultative anaerobe. Membranous, thin coherent growth like a membrane. Subsurface growth. Turbidity. A common name for turbidity is cloudiness. Bacterial growth in broth makes the broth turbid or cloudy at A levels in the tube from top to bottom. Granular, small particles. Flocculent, small masses of growth of various shapes floating in the culture fluid. Some bacteria form clusters of growth that are either floating in the broth or stuck to sides of the tube at every level. From top to bottom, these bacteria correlate with facultative anaerobe. Flaky, large particles. In liquid media, some bacteria grow diffusely, producing uniform clouding, whereas others look very granular. Layering of growth at the top, center, or bottom of a broth tube reveals something of the organism's oxygen requirements. Amount of growth. A. None. B. Scanty. C. Moderate. D. Abundant. Bacterial growth in broths is indicated by the development of a cloudy appearance if the newly inoculated broth looks cloudy at the start. You will have no way to determine if this is due to bacteria. Sediment, none, or A. Granular, small granules, B. Flocculent, small masses of growth of various shapes, C. Flaky, separate flakes, The viscid sediment rises as a coherent swirl. Some organisms will diffuse uniformly throughout the broth, some will sink to the bottom and form a sediment, some will grow in clumps, producing a flocculent growth. These are the examples of some of bacteria and its characteristics.
What is agar? Agar, a gelatin-like substance extracted from red algae, is commonly used to culture microorganisms. Various nutrients are added to agar to enhance the growth of bacteria in either shallow plates or test tubes. When agar media is placed in test tubes, it is in liquid form. The test tubes are placed at an angle to cool and congeal, creating a slanted surface or an agar slant. The first characteristic is amount of growth, and this has 4 types. The first one is scanty or slight, or this can also mean a small amount of growth. The second is the moderate, and the third is abundant or the large amount. And lastly, none. Second characteristic is color of colony. Some bacteria are chromogenic, meaning they can produce some characteristic color or pigment that is useful as a means of identification, and this can be yellow, blue, or green, red, and pink. The third characteristic is consistency, and this has 4 types: butyrous, viscid, membranous, and brittle. As you can see in the picture, Bacillus subtilis is identified as butyrous, which is butter-like. Second is the Escherichia coli, which is membranous, meaning thin, coherent-like membrane, consistent. And third is the Micrococcus luteus, which is viscid, meaning growth follows the needle when withdrawn. And the last is the Mycobacterium flay, which is brittle, meaning dry or friable.
Number 4 in examination of agar slant culture is optical characteristics. For optical characteristics, there are 7 to consider: opaque, translucent, opalescent, iridescent, fluorescent, dull, and glistening. For opaque, no light passes through, and objects behind the test tube wouldn't be seen. Translucent, some light can pass through, but you couldn't fully see the objects on the other side. For opalescent, the results would have an opal-like color. In iridescent, it will exhibit changing rainbow colors in reflected light. Fluorescent will have one color be reflected by light and another by transmitted light. This is used in the analysis of E. coli bacteria because the luminescent signals from metabolites will be an indicator of the vitality of the bacteria. Then there is the dull, which do not have any gloss, and for the glistening, where there is a glossy-like appearance. Number 5 is the form. There are 6 types of form: filiform, spreading, echinulate, beaded, rhizoid, and arborescent. In filiform or unieffuse, colonies grow along the line of inoculation. For spreading, the growth of the colonies extends beyond the line of inoculation. For echinulate, it has jagged margins. For beaded, there are separate or confluent colonies at about the half end of the inoculation. For rhizoid, the colonies are irregular with a branching root appearance. For arborescent, the colonies have tree-like and branched appearance.